will either make you very happy or very sad over the next 15 minutes. So first, in general, phylogenetics forms the backbone of comparative sequence analysis. Any type of thing that we do that's comparing different species to each other, involving genes, determining evolutionary relationships, comparing phenotypes, we basically need a phylogenetic structure in order to properly control for shared ancestry, right? So a key thing is we need to make sure that our inferences in phylogenetics are reliable. How can we do this? We can adopt robust statistical approaches, pick data wisely, analyze data that has reasonable levels of signal, and the one we're going to focus on today is employ an evolutionary model that is well fit to your data. So, quick background, phylogenetic models, for the most part, this is not everything, but the standard ones that you're going to come across, model sequence evolution as a Markov process, which specifies the probability of transitioning, evolving among different states, depending on what kind of data you're working with. These processes are memoryless, which is to say they don't care what you were in the past, they just care what you are now. So if you imagine these nucleotides evolving over time, so here there's 2 times we change from an A to a T, to a G. Both of those changes are described by the exact same probability, even though there was a T right here and a C right here. Briefly, this is kind of your standard nucleotide type of model. It's a 4x4 matrix describing from one state to another state. And we have these 2 main components in our matrix, which are the substitution rates and equilibrium frequencies of what we're transitioning towards. So this is a nucleotide model. It's 4x4. I can show it to you on a screen. A protein model, which is mostly what I'm talking about today, it's going to be 20x20. A little bit harder to show on a screen, but that's where we're headed up next. Okay. Importantly, there are hundreds of ways to parameterize these models to come up with different substitution rates, different frequencies, and many, many different levels that go into this. So something that you're always encountered with when you have to build a tree is, what model should I use? There's so many out there to choose from. How do you do that in a consistent way? So first, I want to talk about this quote from Berneman Anderson. So this is a book that we've all read, right? Okay. So a model is a simplification or approximation of reality and hence will not reflect all of reality. Very famous quote. Fox noted that all models are wrong, but some are useful. While a model can never be, quote, truth, emphasis added by me, a model might be ranked from very useful to somewhat useful to finally essentially useless. And this is sort of the backdrop of how we generally choose our models. We rank them in terms of what we think is most useful to least useful. So how do we do this? I'm not going to get into the details too much, happy to talk more after. But we oftentimes, not always, asterisk, but oftentimes we use a technique called relative model selection, where we have a criteria, something like AIC or BIC, which are quantities that you can calculate from likelihood and from number of parameters, and we rank models according to these criteria. What these criteria tell us is how closely the model fits the data, but they also penalize for overfitting. What I mean by that, if you look at these 3 images below, this blue line modeling the data is sort of a just right model. This one is underfitting the data. It doesn't sufficiently capture the variation. This one's probably overfitting it. You can imagine if I tossed more orange points in the situation, they may not exactly follow that jumpy up and down pattern. It's a bit too tailored to the data set at hand. So things like AIC and BIC are going to provide us with kind of a rank for how can we find that nice little Goldilocks area of model fit to our data. Model selection has come to be strongly recommended, if not sort of dogmatically required for any kind of phylogenetic analysis. It debuted with this seminal model test paper, over 20,000 citations and many, many other citations for all these different methods that are out there for how you can rank your models according to what's out there. Very, very common. Now, this is something that I want to point out. So this is an example. If anyone's ever studied HIV, you may be familiar. There's an HIV database online. There's many, many features of this database to help you analyze your data. But it's got this one feature called find model. It's got this sentence in there, boxed in red, which I've enlarged here. Find model analyzes your alignment to see which phylogenetic model best describes your data. This model can then be used to generate a better tree, is how they describe it. There's no justification for that. This is an assumption that has been flying around for a long time, but there's really no guarantee that model fit, especially relative model fit, which is just ranking what's available, not determining what is absolutely the best, there's no guarantee that that's going to give you, quote, the right tree, more likely than anything else. So another take when thinking about model selection comes just from the many words of Andrew Gellman. We do not like to ask, is our model true or false? Since probability models in most data analysis will not be perfectly true. The more relevant question is, do the model's deficiencies have a noticeable effect on the substantive inferences? So what I'm really interested in is actually looking at the empirical results. What happens if I use a quote, terrible model? What happens if I use the, quote, best model? Are there actually tangible differences that come out at the end of the day? So again, the nearly universal assumption that the best fitting model produces the best, i.e. most accurate tree, this is largely untested. I will point out there's some recent studies that came out in the past couple months that have spoken to this a little bit, so it's not just me, but we're all getting on the bandwagon now. So in this study I asked, what is the relationship between model fit and inference accuracy, specifically for empirical models of protein evolution? The reason that's important is because proteins do not evolve. DNA evolves. Sometimes this has consequences for the proteins that come out of the DNA, but fundamentally, proteins don't evolve. So all these models are purely phenomenological. They can only describe patterns of evolution, not the evolutionary process itself. So as a consequence, the way that these models tend to be made are you train them on giant data sets. You get hundreds or thousands of alignments and trees. You use maximum likelihood to fit models. You end up, this is a JTT model, so you end up with these kind of different rates for different amino acids that can be represented like a bubble plot for substitution rates among them. Now, the reason that we've been doing this and training them a priori is because these models have 189 free parameters, which is a lot of free parameters. So this scares us a lot. We think we're going to overfit the data, so we can't estimate the parameters from the data the way that we might for a nucleotide model, because 189 is just too many. So we use these fixed, pre-trained models for protein analysis. So here's another thing. So what I'm going to do is I'm going to test this assumption about accuracy and model fit. Now, when we want to test assumption, when we want to really test the truth of something, simulation provides a really, really powerful strategy because we know what the true answer is. However, simulations can also be highly tautological with following circular logic. So for example, let's say I want to know something about model X. Maybe I simulate under model X, I use inference with model X for whatever I'm interested in, then I compare to true values and I say, wow, model X works really well. Well, of course it worked really well. Your model and your data fully match. This cannot, but when the model matches the generative process entirely, we have no idea how the model behaves when data is misspecified. All natural sequence data violate our models. Models cannot fully capture reality, so there's always going to be a divide in there. So what I'm going to present to you is a simulation strategy that can get away from this tautology so we can separate the generative process from the inference process to get a bit more robust results. So what I do here is I actually perform simulation with a codon level model. It's called the mutation selection model. Basically, it considers at each site in a protein, there is a specific set of amino acid preferences at that site in the protein and a global set of mutation rates. So you're actually taking into account the fact that there's different evolutionary constraints operating across the protein. You don't have a global matrix. On the other hand, for inference, I'm going to use these protein exchangeability models, which have nothing to do with the site-specific nature of the simulation. In the interest of time, I'm not going to get into the model too much, but happy to talk later. So what is my approach? I did 20 replicate simulations for each protein tree combination you see below. So what is this here? This is a quick little table of 3 sets of parameterizations that I'm using for simulation that are derived from deep mutational scanning data, so from experimental data run on 3 different proteins, flu nucleoprotein, flu hemagglutinin, and HIV envelope protein. And I'm simulating along 8 different trees. If anyone is really into phylogenetics, some of these trees may be familiar to you as trees that have caused various levels of conflict in the field by being difficult to resolve the relationships among. Okay, and again, since simulation was done at the codon level, I translated everything to amino acids before proceeding. I ran standard model selection on all alignments, which looks among 168 different parameterizations of amino acid models to determine which one best fits the data. Then I inferred phylogenies for each alignment. Models M1 through 5, hold on, the Jukes-Cantor model, which is our simple equal rates model that nobody would ever use because it's too simple, it underfits the data, not worth our time. And then a GTR model, which estimates all 189 exchangeabilities, and many people will tell you, don't use this, it overfits your data. So we're underfitting, overfitting, and then this right here. So what's that? Oh yes, and then I calculate Robinson-Gould's distance to the true tree. And in general, interestingly, results are actually pretty consistent across all simulations. So I'm just going to show you a subset of results for the rest of the talk, just from the mammal tree and the Lassivirus tree simulated with hemagglutinin parameterization. So that M1 through 5, what is it? So what I did, for each simulated alignment, I shoved it through model selection. What you get there is 100, each point in this distribution represents the BIC for one of the models, for each model that was tested. I then chose the models that were the best fitting model, M1, the worst fitting model, M5, and M2, 3, 4. These represent the quantiles of the full distribution. So we have a clear way of looking for each alignment, what are the 5 models that are equally spread out across the whole data set, ranging from best fitting to worst fitting. I also have Jukes-Cantor, which tended to fall right about here for simulation, so between M4 and M5. And interestingly, the GTR model. The GTR model for the simulated data was actually the best fitting to all the data, which is probably because the simulations were pretty informative. So, first thing, Robinson-Gould's distance to the true tree. What I'm showing you here along the x-axis, each of the models, each point here represents a simulation under one of these 2 conditions. The thing that I want you to notice is that with the exception of M5, Robinson-Gould distance is pretty constant across everything. So again, this is your worst fitting model, oh gosh, I'm in trouble here, marginally significant in terms of these other values, but only about, overall across all data sets, 3% farther away compared to the best model. Only 3% farther away. Okay, so in the interest of time, sorry guys, we're gonna skip ahead a little bit. I also looked at bootstrap values for this, so, gosh, we're really running out here. Okay, sure, why not? So I used ultra-fast bootstrap for this, which is part of IQ tree. These have a slightly different interpretation. Above 95, well supported. Below 95, poorly supported. What you can see, if the node is present in the true tree, we're mostly above the line. Not present in the true tree, mostly above the line. This is, for a single replicate alignment, it's pretty constant throughout, even Jukes-Cantor, even GTR. So we see a lot of constants in the entire tree. And what I'll tell you, just skipping ahead, looking at false positive accurate nodes, the point is, false positive nodes, no matter what model you're using, you're always getting some proportion of nodes that are incorrect on your inference. You're always getting roughly the same portion of nodes that are correctly classified in your inference, accurate nodes. A bit dip for M5 and a bit of a dip for M5, but it's not statistically significant. I also ran Topology Test to formally test, are there differences for each alignment and the 8 trees associated, the 7 trees that we inferred, and the 1 true tree? Do they fall within the confidence set of the best fitting model? Because there's no true model that we can use for any of these trees because the simulation strategy is totally distinct. But so we're comparing to basically the M1 model, what would normally be used in model selection. What we actually find is trees are all basically fully consistent with the M1 model. So even though you can take your worst fitting model, you can take a middle fitting model, delta BIC 3000, your results are all falling within the confidence set of the best fitting model to your data. So sorry, I wrote a talk that was a little bit too long, but we'll just wrap it up there. My conclusion is basically GTR is not actually that bad. Go for it. And some results coming up later. Preprints coming out soon. Don't worry about it. There we go. Better fitting models do not systematically produce better trees. Generally, nodes that differ from the true tree have lower support, but all models create trees with highly supported incorrect nodes. All models, your best model, your worst model, your middle of the road model. Are models all equally right? Are they all equally wrong? I have no idea. The GTR model performs on par with your best fitting model in spite of overfitting considerations. Caveat, I don't consider mixture models or multi-gene models. That's coming up next because those are more complex. And I know, rush through this a little bit, but please, I'd love to talk to you. Twitter, email, IE. Preprint forthcoming. Thanks to everyone at Dalhousie University who I visited a couple months ago, and they were super helpful in talks. Sorry for going over. Thank you for your patience.